Well, thank you so much for inviting me and for the kind introduction. As Leon mentioned, I've spent a couple of years in the Boston area at the Broad Institute when I was a postdoctoral fellow in Aviv Regev's lab. So it's good to be back, even remotely. And today I'm going to focus on a particular study that I was asked to present, where Aviv and I put forward this concept of multicellular programs. And I actually started this study when I was at the very end of my postdoc, and since became a PI and established my own lab. And so, as you can imagine, this was a big shift in my research. And I'm going to try in this talk to also give you a little bit of a flavor of the type of approaches that we're taking and some of the new directions that I think will also provide the broader context for this specific contribution and study. Okay, so the talk will have 3 parts. I'm going to start with an introduction to multicellular biology. I don't think there is a subfield that is yet called multicellular biology, but I think there might be a need. Also, looking at the talks that you host in this seminar, and then also tell you about our approach more generally. Most of the talk will focus on the study that we published a few months ago, where we introduced the concept of multicellular programs and also developed, as far as I know, the first dimensionality reduction method that allows us to extract information about multicellular programs from either single cell or spatial transcriptomics data. And I'm going to walk you through the concept, the method, and the different applications. And lastly, I'm going to share with you some of the new directions that we're taking to uncover mechanisms of multicellular processes and to start thinking of ways to engineer multicellular circuits. So taking a step back, I want to put this in the framework and in the context of the genotype to phenotype mapping, because I think almost no matter where you are in the range of biomedical research or biology, this is a fundamental question and problem that we're all trying to address in one way or another. So the need to link between specific genes to complex phenotypes is a major challenge. And we've made progress in this direction with, for example, genome-wide association studies that allow us to link between genetic variation and different complex phenotypes. But one of the limitations that we face is that most phenotypes involve the collective behavior of different genes. And so this one-to-one mapping that we were hoping to have is not really supported by the data. Most phenotypes are more complex than that. And in addition, nature doesn't like doing experiments. So we often see variation that is in the non-coding gene, which is more difficult to interpret, and variation that does not have a major impact, where that's a good thing, because otherwise diseases would have been more prevalent, but it makes it difficult to study connections. Now, fortunately, we now have systems as CRISPR that allow us to probe different genes and to measure the effect that they have on well-defined phenotypes. And these are usually phenotypes at the cellular level, whether it's fitness under different conditions or a specific molecular readout. This is great, but a lot happens in between when different molecules within a cell interact and when different cells start impacting one another, and each cell by itself is a function both of its genes and its environment. A lot of the processes propagate to impact multiple cell types, multiple cells in the body, and only then are generating the final phenotype that we care about. And so, unfortunately, we still don't have today high throughput, quantitative, engineering-based approaches to tease apart molecular mechanisms at scale in this manner. And however, I think we are in a very unique point in time where we can start putting together different domains of knowledge and different technologies that we have today in order to make headway in that direction. And if we'll be able to do that, I think it can fundamentally change the way that we study biology, the way that we understand biological systems, as well as diseases. And this will have implications for diagnostics, treatment, and prevention. So in my lab, we hope to contribute to establishing these types of systems. And we're particularly interested in the immune system, as Liang mentioned, and particularly in the context of the cancer-immune interplay. Now, the immune system is a phenomenal system, and it's a beautiful example of how different cells can cooperate and make decisions together and form very complex functions, even memory. And the way that they do this is in part by roaming the body, circulating the body, collecting information, and they really need to be in the right place at the right time. And so a lot of pathologies, and this was also something that I focused on during my postdoctoral work, a lot of pathologies begin when immune cells are not located in the proper tissue, or do not reach the tissue, or do not reach the tumor. And so in my lab, we're combining spatial transcriptomics, high content CRISPR screens, and machine learning in order to identify regulators that control spatial distribution of different immune cells, both within and across tissues. And we're also taking this uber reductionist approach. We're going to talk a lot about multicellular programs today, but at the end of the day, the building blocks are one-on-one cell-cell interactions. And so to understand the mechanism, we are taking, going back to these building blocks. One of the things that I showed during my postdoctoral work was that when we look at T cells and cancer cells, the interactions between them are regulated and controlled by different molecules, both on the T cell side and on the cancer cell side. And so today we have more powerful technologies that will allow us to decode both the regulators that control the formation of these interactions, for example, the formation of immune synapses, as well as the outcome of these interactions at scale across different systems and settings. Now, I describe these two processes as if they're independent, but actually one of the things that we showed in melanoma in our paper in Cell 2018 was that these two processes are in fact intertwined and tightly linked to one another. And so now with this more mechanistic information, we're aiming to address one of the more challenging questions, which will be also at the center of this talk, which is to understand emerging properties when different cells, many, many millions of cells, both within the tissue and across the body, are impacting one another and creating these systematic effects. And one of the incentives that drew me to this was the work that we've done in the cancer immunology setting, where we demonstrated that in certain patients, there is both, and not quite a lot of patients actually, in melanoma, there is both a need and potential to target different genes across different cell types in order to unleash effective anti-tumor immune responses. And so if we understand these nonlinear combinatorial effects across different cells, we'll be able to perhaps understand very systematic effects and lead to new types of interventions. And so with that in mind, our objective, the objectives of my lab are twofold, and it's sort of like two sides of the same coin, the way I see it. One is to tease apart mechanisms, molecular mechanisms of multicellular processes at scale with engineering quantitative-based approach. As Liang mentioned, I come from a fairly CS background, mostly computational biology. Today I have a wet lab, and I think this is going to help us advance in this direction. And then the other side is to start thinking of cells or groups of cells as little machines that we can engineer in order to probe or create new functions. And one of the neat things about immune cells is that we can take them out of the body, we can modify them ex vivo, and then we can bring them back. And that makes it so much easier to start studying these processes, both to understand how they work and when thinking of interventions, clinical interventions, etc. So the way that we do this is, as I mentioned, we're fairly multidisciplinary, and the way that we go about it is by creating this hybrid in vitro, in vivo, in silico framework, where machine learning models are an integral part of our experimental design. And they also are an hypothesis generating tool. Usually they're far less biased than we are. So that's the exciting part, because it kind of pushes us to try new directions, to look at things that nobody else is exploring. And that's a lot of fun. And then our experimental models and patient-focused studies are designed in a way that allows us to obtain multi-level layered readouts and data that we can then use to first validate these predictions and gain insights to how things actually work, not only in theory, but also we can feed this data back to the machine learning models in order to further train them. So it creates this reciprocal flow of information, which I think is really important. And many people are doing this in the field also through very large collaborations, and also in this context in a fairly multidisciplinary lab. Now, another axis of integration in my lab is combining patient-focused studies and perturbation-based studies, where we can do massively parallel experiments and follow up on what we find in patients. We already see that by looking at tracking multicellular dynamics in patients, this allows us to broaden the scope of immunomodulating mechanisms and interventions that we consider and target. These functional studies are, I think, really a game changer, and I'm going to talk about it a little bit at the end. But collectively, this generates these multimodal data, and the machine learning is in many ways the glue to that puts everything into a unified system. And that won't be possible without this awesome team. These are the members of my lab, and just a lot of fun to bring in people with different expertise that work together. At the end of the day, it's really about communication. It's really about, you know, the ability to work together, and just exciting to pursue these directions together. And our scope is fairly large, as written here, and I alluded to this as well. So with that in mind, I think this puts the study that I'm going to present in context. And now I want to talk a little bit about multicellular programs and what they mean. What are some of the applications of this approach? So again, this is a study that we recently published together with Aviv Legev, and the idea here was fairly simple. I can't see the people who are listening, but I guess you're probably familiar with the concept of gene modules, right? We've learned a lot from being able to describe gene programs or gene modules or gene signature. They have different names, but they basically mean a similar thing or the same thing, which are situations where different genes within a cell are co-expressed or co-regulated. And that's the reason that dimensionality reduction is so useful in biology, because genes usually work together, and we can see this in their transcriptome. So some time ago, Aviv Regev and I set out to test whether gene modules also can span multiple cell types. So these are situations that we hypothesized that different genes across different cell types will be co-regulated, perhaps in a similar way, because these different cell types are exposed to the same environment, responding to the same signal, or maybe they're impacting one another. So again, these are different genes across different cell types that will be co-regulated and co-expressed. And so we went on and developed a dimensionality reduction method that allows us to identify these types of gene modules, but it still wasn't clear to us whether we're going to find strong patterns. But it turns out when we started exploring this, that multicellular programs, as we called these patterns, or MCPs, are basically everywhere, wherever we looked. We found them in the brain, we found them in tumor microenvironment, we found them in inflammatory bowel disease, we found them in single cell data and in spatial data. And the interesting thing about these MCPs is that they explain variation in complex phenotypes. So now if you regress out the MCP information, the residuals can no longer explain, at least in those settings that in those data sets, they were not able to explain these complex phenotypes. They also provided insights to genetic risk loci and to environmental factors that predispose to disease. And I'm gonna show these examples in a little bit. Now, another neat application of MCPs is that they can now help us recover the tissue structure and the generative process that leads to different tissue structures. So, for example, if we now, when we trained our dimensionality reduction on a certain data set, if you give me the transcriptional information of an unseen cell from the same context, I can tell you which environment it came from, and I can recover which cells were next to each other, etc. And I'm going to show that as well. Okay, so I want to go a little bit deeper into the methodology itself. I know that this is a fairly computational seminar, so I thought it will be nice to explain how it works. And to do this, I want to talk about the notion of a shared queue. So the assumption here, the hypothesis here, is that multicellular programs can result from different cell types that are exposed to the same queue. And that queue can be an environmental queue or an intrinsic queue. And I'm gonna explain what I mean by that in a minute. But as you can see here in this figure, this is from our paper, this queue can impact the different cells in the same way, or it can impact them in different ways. And we actually see that this is usually what's happening. And there are also situations where it can impact one cell type and then propagate to impact the other cell types. So what is this shared queue? We mostly think about this in terms of environmental factors that are different cells that reside next to each other. They're going to be exposed to the same signal, and can be metabolite or a physical pressure, a combination of cytokines, all sorts of things that we don't measure. But it can also be an intrinsic queue. For example, all the cells in our body share a certain genetic variants, right? And if we look at cells from a different individual, they're going to share a different set of genetic variants. Also, all the cells in our body age. And so that's another shared queue that they all have, but it's intrinsic. So just keep that in mind. So with that in mind, we're now going to identify MCPs using the method that we developed called dialogue that has two steps to it. The first is a multi-view penalized matrix decomposition or a sparse canonical correlation analysis. That's multi-view. We're going to talk about it in a minute. And the idea here is that we refer to different cell types as if there are different representation of the same thing, of the same cue that we cannot see, or the same environment. And we're then proceeding with a multi-level statistical model that allows us to account for different confounders and account for the hierarchy in our data. So different cells come from different niches or sample and different individuals, different group. We really need to account for these interdependencies. Okay, so let's zoom further to the first step of the multi-view penalized matrix decomposition. And now let's assume that we're looking just at one cell type. You have the different cells, the different features. These can be different genes in your original data. And say for just for the sake of the example, that we know we have a value measured of that shared cue that I was just talking about. Well, in this case, we can just solve a simple regression problem to identify the different weights on the original features that will give a meta feature that will represent that property, right? That shared property. So now let's assume that we have a different cell type and we can repeat the same process. We're probably going to get a different vector of weights, especially if the cue is impacting the cells in a different way. But we can recover that. The only problem is that we don't have information on that latent variable. But what we can do is solve a different problem, which is to constrain the two to be as similar as possible. And that gives us the opportunity to recover latent features from our data. And we're going to call these weights, or the genes that are most dominant in these vectors, as the multicellular programs. And of course, this was just an example to show this for two cell types, but we can do this for as many cell types as we want, and we can identify multiple multicellular programs. And this is just the way that it is formulated mathematically. And then, as I mentioned, it's also important to account for the single cell distributions and for different confounding factors. And so we plug it into a multi-level model where we have different nested levels, and we can account for the dependencies, and we can account for the hierarchy and structure of our data. So this is demonstrated here. So now that we applied a dialogue to different data sets, let me tell you a little bit about what we found and how they look like. So this is an example from lung cancer. This is spatial data with CosMx, or SMI, that was applied to 8 non-cell lung cancer tumors, where they measured almost 1,000 genes across 800 single cells in the tissue, in the intact tissue. So here you can see the tissue section where cells are annotated and colored according to the different cell types. And here you can see a multicellular programs that we found in lung cancer spanning CD4 T cells, macrophages, and fibroblasts. And we highlight in green, red, and blue the cells that overexpress the cell type specific component of each MCP. And you can see how they are nicely co-localized in the tissue, which means that while if we were to do a standard dimensionality reduction, we would probably assume that these different programs or these different gene modules are independent, but in fact they are not. And so when we zoom further into the MCPs that we identified in lung cancer, we see that they capture well-established immune evasion mechanisms. And it's funny because I just took this picture from a wonderful review that was published back in 2015, where they did sort of a survey of the different immune suppression mechanisms in the tumor microenvironment. And this MCP, in a completely unsupervised manner, managed to recover those connections. And so it demonstrates the IL-1 signaling with both the receptor and the ligand expressed in macrophages and cancer-associated fibroblasts. It shows that different cells respond to interferon type 1 and type 2 in different ways. And one of the genes that is probably responding to interferons is IDO, which is known to metabolically shift the tumor microenvironment to repress T cell proliferation and to induce the proliferation and the persistence of regulatory T cells. We also see genes as ICAM1, which can recruit T cells and improve their adhesion when they are recruited into the tumor microenvironment. However, we also find VEGF-A, which is known to essentially lead to a differential recruitment of regulatory T cells by inducing the expression of Fas ligand on the endothelial cells. And so this is just an example of the type of things that we can recover. Now, MCPs and dialogue can also tell us something about the directionality of impact and of communication, if you'd like, between different cells and the tissue. So this is an example from a totally different context, from the hippocampus of mice. And here we can see this is just a layer of astrocytes demonstrating MCP1 as shown here in the color code. And what we can see that in this layer, there are certain astrocytes that communicated or are impacted by neurons in this space, while MCP number 2 is going to show us something very different. And these MCPs are orthogonal. And so there are different cells in the same layer that are communicating with neurons in different directions. Now, I'm not a neurobiologist, but for me, this was interesting. Now, as I mentioned before, we can also now use this information as a sort of, you can think about it sort of like a linear autoencoder of the tissue structure. So now that we have information about MCPs, we can, and this is shown here from Murfish data from the mouse hypothalamus, where we show that we can now, given gene expression information from one cell, we can predict on unseen data the expression of the nearby cells from different cell types. We can also, given that we have a pair of cells, we can predict whether or not they are neighbors. And this is shown here. You can see the performances of the MCP compared to just using the gene expression by itself or using the PCs. There has been previous work of I think it was Mornington and Nature published in 2019, if I'm not mistaken, where they showed beautifully how the gene expression by itself can be used to predict whether cells are next to each other, just because they're going to have a more similar gene expression. And so we tested this here, and you can see the performance of dialogue in comparison. Another thing that we can do, and this is based on single cell data, is to spike in cells that were collected from the same patient, but from a different sample, and use this information to this contaminated data to train dialogue and see if it can spot the outsiders. And it turns out that it can. So, this could potentially be used to identify newcomers or cells that just infiltrated into the tissue. But here we can clearly see that when we simulate data in this manner, we're able to spot the outsiders. And this is just stratified for different types of contaminations and different types of cell types from colon biopsies, single cell data that we tested this on. Now, you probably ask yourselves, what about other methods? Spatial biology is a field that's evolving, and there have been previous methods that focus on spatial signatures or spatial distributions of gene expression. One beautiful study from Zhao et al. That was published back in 2018 used hidden Markov random fields in order to formulate spatial data as a graph and identify spatial domains in the tissue. And that's a beautiful study. However, it's assuming that different cell types are responding in the same way to their environment. This is an intrinsic property of the model that they used. And so perhaps it's not surprising that when we examine the domain-specific signatures that they identify, they do not show the property that we show with dialogue, where the expression of the different cell type specific components of the gene module are co-regulated and co-expressed spatially. And so while it captures domain-specific gene expression, it doesn't capture multicellular programs, at least as we define them. So you can see this here. Okay, so far we talked about spatial variation, and there's a lot that we can learn from spatial data, and the statistical power, of course, is much greater than looking across different individuals. But in fact, there are many different types of variation that can be extremely meaningful, especially when we're thinking of studying diseases. And there are different levels of variation that collectively can be integrated. So what I mean by that, you can think of multicellular programs that recapitulate variation across different anatomical sites, different tissues, longitudinal data where, for example, a sample is being collected across different time points, and also variation across different individuals. So let me show you a few examples that follow this line. And we actually started thinking about this when we applied dialogue to merfish data from the mouse hypothalamus. We found different spatial multicellular programs, which are shown here. But we also found that the most dominant multicellular program that we identified captured variation in the behavior of the mice. So some mice were considered naive, and others had different experiences. And the MCP, in a, again, completely unsupervised manner, managed to capture it. Now, I remind you, this data includes only 130 genes. So perhaps that explains why we couldn't be even more granular. It is curious to see whether we're going to identify something that's behavior specific MCPs if we, once we have larger cohorts, and we're certainly heading in that direction in terms of the technology. But this encouraged us to look more broadly at this property. And what we found, we started applying dialogue to different single cell data sets. So I want to show you a few examples that demonstrates its value in this context. When we applied it to melanoma tumors, we found a multicellular program across CD8, CD4 T cells, macrophages, and B cells. And here we're showing this as a heat map where each row corresponds to a different patient, a different sample from a different patient, and the different columns correspond to different genes from different cell types, which are part of this MCP. And what this captured was a multicellular program that links T cell exhaustion and macrophage polarization, as well as the repression of ApoE. And interestingly, there is beautiful work from the group of Svahatabazoy where they showed that ApoE repression and germline variants lead to resistance to PD-1 blockade and immune suppression via myeloid-derived suppressor cells. And we actually have some more mechanistic data suggesting that some of the effects might be directly on the T cells. And here you can see that this multicellular program that we identified is also tightly linked to the response of these patients to immune checkpoint blockade that they were later treated with. So this is one example where we showed the connection. We started showing the connection to treatment response. Another example comes from inflammatory bowel disease or ulcerative colitis, where we applied dialogue to single cell data from colon biopsies. And there we found a multicellular program that was linked to genetic risk loci. It was enriched with genetic risk loci, and perhaps allows a way to assign different genetic variation predisposing individuals to disease to particular cell types. And that's, of course, a major challenge. So here you can see the multicellular program that we identified in IBD. You can see the cases and controls. And one of the interesting properties of this program is that it is just as dominant both in the inflamed and non-inflamed samples that were collected from the patients compared to controls. And so while we cannot, it's not manifested at the clinical level in terms of the histology and the pathology, it is at the molecular level, and it allows us to distinguish between healthy individuals and those with IBD. And we show this also in an independent unseen data set, which was collected across dozens of patients, and show that the ulcerative colitis MCP that we identified can distinguish between patients without individuals without ulcerative colitis and those that suffer from the disease. And it can also distinguish, and you can see the AUC here, it can also predict, based on pretreatment biopsies, the response to the therapy, which is TNF inhibition. And perhaps if we, as you can see, there is pre and post samples matched from the same samples, it doesn't seem like this treatment is repressing the program, but perhaps it could be a biomarker for drug development in the future. And the last example that I want to show you comes from Alzheimer's disease. This is application of dialogue to single nucleus data from brain autopsies that were collected from Alzheimer's disease patients and individuals which were neurologically normal. And what we found there were two different multicellular programs that are both associated with Alzheimer's disease. And so this is just an hypothesis at this point, but potentially we can use this information to identify different subtypes of diseases or different molecular manifestations that lead to converging phenotypes, which are in fact can be distinguished. One of the MCPs, and these two are shown here across the different cell types, one of the MCPs was enriched with stress genes with the repression of synaptic signaling, which makes a lot of sense. And the other MCP was highly enriched with genes that bind to metal and zinc ions. And now this is not my field, but so I was not aware of it until recently, but it turns out that if you look at the plaques in the brain and you stain them, and then you stain for zinc, this is what you see. And so perhaps this MCP was able to recover this latent property of the tissue related to the abundance of zinc, and that could potentially lead to the pathology that we eventually see. And lastly, we turn to examine whether this is linked to disease predisposition in Alzheimer's disease. We know that Alzheimer's disease is a function of age. We rarely see Alzheimer's disease below the age of 50. And so we used data from hundreds of individuals, which were neurologically normal. And what we found was that these two MCPs are upregulated, are increasing, and you can see this here, in different parts of the brain in a nonlinear fashion as a function of the age of the individuals, and this was from autopsies. So these were just a collection of a few applications of dialogue. We tried to show this in a variety of settings to gain insights and to see whether this is a generalizable property. We have a tool online, it's on GitHub. Feel free to give it a try, tell us what you think, and hopefully you'll find something interesting, or that this will inspire you to develop other methods along the same lines. And before I summarize summarize, I want to say a few words about the new directions that we're taking to go from multicellular programs to mechanisms and to interventions, because I think that this concept is very useful of MCPs, but one of the challenges will be, number one, to be able to identify those across different settings, and number two, to be able to tease them apart and understand the cause and effect, so that we'll be able to have, concrete leads for interventions and to understand the mechanism. And so to do this, we, as I mentioned before, I think both patient-focused studies as well as perturbation-focused studies, the integration between them will be very important. So I want to show you some directions along these lines. And when I started my lab, we tried out many different spatial transcriptomics technologies, invested a lot of our efforts in that. And I'm very glad to say that I think the last year has been a major step forward in terms of spatial transcriptomics for a couple of reasons. First, we can now have subcellular resolution. We can measure an increasing number of genes in situ. We're talking about hundreds to thousands of genes. And we can also start using archival tissue samples. And all of these things are things that were not possible with single cell genomics because the tissue is dissociated. So very difficult to study how different cells impact one another because we don't know where they reside, but also because we weren't able to design studies across many different patients and to design them to eliminate confounder and to address specific questions of interest. And so one of the reasons my incentives to study what's happening in patients is because I think, number one, it can broaden the spectrum of our view and our perspective. A lot of our models are not really capturing what is happening in a patient. And also, I think that there is value in going from reducing the search space based on associations and then following up on that. So I want to show you just one brief example, and this is from my postdoctoral work on melanoma, where we set out to identify the connection between cell states and tissue composition, particularly focusing on T cell infiltration and recruitment. So one of the things that we know for sure is that for any type of T cell mediated killing or any type of T cell focused immunotherapy to work, we need the T cells to be properly recruited and to infiltrate into the tissue. And so by looking at cold tumors, which are immune deprived, we can gain insights to what might be preventing T cells from being recruited. And this is exactly what we've done. This is all published, so I won't elaborate too much on that, also due to lack of time. But we applied different types of inference approaches on single cell and bulk data, identified a transcriptional program that is unique to malignant cells and cold tumors. And we showed that we can use this information to predict responses to immune checkpoint blockade across large populations of patients, and that we can use this information to also predict interventions that will repress this transcriptional program that characterizes malignant cells and cold tumors in order to unleash effective anti-tumor immune responses in a variety of ways. One of them was using CDK4 and 6 inhibitors. And so now with spatial data, what we can do, we can directly measure this, and we can examine whether there are specific features in malignant cells that will be associated spatially with the infiltration and recruitment of T cells or other immune cells in the tissue microenvironment. And so this is exactly what we did in the new spatial data that we collected in ovarian cancer, and was really remarkable to see that despite the heterogeneity across patients, we find that the connection between where T cells are and the expression of specific genes in the malignant cells So the T cells are shown here in black, and all the other cells are malignant cells, which are colored according to this signature that we identified, this program. We see this over and over again across multiple patients. And so we now use Perturb-seq to identify latent regulators of this program and are testing this in cancer lymphocyte models. And we're very excited about this because it both corroborates our previous findings, but there are also multiple novel regulators that we find with this approach, and particularly in ovarian cancer, where there is a huge need to make progress. So I just want to say a few words about Perturb-seq, because I think that high content CRISPR screens will help us delineate the mechanisms of both cellular and multicellular processes. Perturb-seq is a method that was co-developed in Aviv Legev's lab and co-developed also in Jonathan Weissman's and Ido Amit's labs, where we can do multiplex pooled screens and then collect information at the single cell level. For example, single cell RNA sequencing, site-seq, attack-seq, etc. And so we're now taking this one step further by establishing systems of high content CRISPR screens with in situ readout, so that we'll be able to measure the impact of a perturbation both on a cell itself and on its microenvironment and its neighbors. I think that this will be an important technology to decode cell environment and cell-cell interactions. And the reason that I love this approach of high content CRISPR screens in general is because they allow us to combine forward and reverse genetics. So forward genetics is a situation where you have a particular phenotype of interest and you want to identify the specific genes that give rise to this phenotype. And that's usually done with traditional CRISPR or GWAS. And reverse genetics is a situation where you have a particular gene and you want to understand its function. And so you do, for example, mechanistic studies, and you can study something more fundamental about the way that things work, and also in terms of combinations. So just to briefly explain why I think that this is so critical and important, and I worked quite a lot on combinatorial effects of genetic interactions when I was a PhD student in Eitan Lupin's lab. So if you think of different perturbations and the different phenotypes that they can lead to, most of the phenotypes require large in vivo experiments per perturbation. So this matrix is going to be extremely sparse. We also know that the perturbation effect on a phenotype is going to be context dependent. We see this with immune checkpoint blockade, right? Some patients are literally cured while others succumb to the disease. And so already we're starting to get to a fairly high search space. And lastly, many phenotypes result from nonlinear combinatorial effects, both within and across cells. And now this is becoming untangible to measure. And so what we propose is to take the beautiful traditional classical biology and apply it to machine learning and the type of data that we have now. So traditionally, we look at a specific gene or protein, we try to understand what it does, and then we link it to phenotype. Now we can use genetic perturbation molecular readouts, embed this in a machine learning approach using transfer learning, etc., and predict a variety of phenotypes, as well as the effect of different combinatorial interventions on different phenotypes. And this can be done using transfer learning, and something that we're actively working on. We're also generating the data that we need in order to train these models using perturbation-based studies with massively parallel experiments and cell engineering using both the genetic and epigenetic perturbations of knockout, activation, inhibition across in vitro, ex vivo, and in vivo models. And on top of these molecular readouts, we also have functional readouts of cell location, cell-cell interactions, and emerging properties. And this is a major team effort within the lab, where different studies are also for us very exciting, each one by itself, and together will form this hopefully a fairly unified view that we'll really look forward to share with the community. Okay, so to summarize, multicellular programs uncover transcriptional regulation across cells. They can explain and predict spatial structure in various tissues. They mark complex phenotypes, and they can help us define potentially. We haven't validated this, but there might be potential to define new disease subtypes and to elucidate both the genetic and environmental factors that can contribute to disease predisposition. As I mentioned, you can apply dialogue both to single cell data and spatial data. There is an intrinsic mechanism in the algorithm itself that will prevent overfitting. So if you don't have enough data, you're not going to get results, or if it doesn't generalize. And with the increase in spatial data, with the increase in single cell data, there might be opportunities to have nonlinear, to identify nonlinear connections across cells, which we haven't done. And lastly, I just want to say that I think that multicellular biology is really the missing link between tissue and cell biology, and that we're in a very, very unique point in time where we can leverage existing and emerging technologies to have the ability to dissect molecular mechanisms at scale. And lastly, if we can decipher the inner workings of multicellular regulation, I think that this will provide inspiration for new ways to engineer not only cells, but engineer collections of cells, and think of ways that we can leverage biology as it works with different cells cooperating with one another. So speaking of cooperation and collaborations, this was a wonderful collaboration with Aviv, which started again when I was a postdoctoral fellow. So she was a wonderful mentor for many years. The melanoma study that I mentioned was in close collaboration with Ben Zal, who's now a PI at Columbia University, and Levi Garraway was my co-mentor. The spatial study that I mentioned is a very close collaboration with Brooke Howitt from Stanford Pathology, and these are the members of my lab. Our funding resources, and thank you very much for your attention.
Great. Thank you so much for the excellent talk, and very exciting talk. We do have one question from the Q&A. So Biju, do you want to ask a question? I'm actually going to unmute you. Okay, I think you can talk now. Just unmute yourself.
Can you hear me?
Yes.
Hi, so it was a nice talk. I just have a curiosity question about MCP. So can it be applied to something like a pseudotime analysis or lineage studies?
Yeah, that's an interesting direction. I think there, as I'm kind of alluded to at the end, the ability to use it for longitudinal data, right, where you could potentially identify MCPs across time, and look at this dynamics. So if you have on top of it also information about lineage tracing or how cells change, you will probably need to formulate the problem more carefully, or add more constraints, or provide the input in a certain way. But I think that's an interesting direction. Is there something particular that you have in mind?
No, this was just a curiosity question.
Okay, great. Any more questions? Please just raise your hand, or just put the question in the Q&A. Let's just give like maybe 1 or 2 minutes for the audience to think about a question. I might ask like a live question. So I think you mentioned about that to use dialogue, we need to have enough number of samples. So I'm not sure, do you have, did you actually do the, study and like say, what's the minimal number of samples we should use to run your tool?
Yeah, a lot of people ask that also on our GitHub. I would say that for single cell data, I wouldn't go below 10 samples, and probably having also data for testing is useful, as always, sort of out of sample testing. Intrinsically, Dialog runs empirical tests to make sure that it's not overfitting to the data. So we take care of that in a way. With spatial transcriptomics, it's not. With spatial data, it's not so much of a problem. Yeah.
Great. That's good to know that. One more question is that do we need to have a good cell type annotation for example, single cell RNA-seq data before we actually use your software?
It depends. It could potentially capture if you don't have sufficient granular annotation of cell types, the MCPs might capture changes in cell type composition. So this is something to keep in mind. For example, if you apply it to all the different T cells, then it might capture differences between CD4, CD8, T regulatory T cells, etc. So I would just keep it in mind. It's also useful to provide the data. So we usually recommend doing some level of a dimensionality reduction before dialogue. So for example, PCA on each cell type, and not doing it collectively on all the cells, because again, then the PCs will capture differences across different cell types and not cell type specific variation, if that makes sense.
Thanks. Any other question? Okay, I think if you have any more questions, you're welcome to contact Dr. Jerby. And thank you so much, Dr. Jerby, for the nice talk and very exciting tools. I think we will try to use that and maybe have some follow-up question with you also. Thank you so much.
Yeah, absolutely. Happy to stay in touch.